Hi there, welcome to this core practical. So this is core practical 3, all about membrane permeability. So this experiment's usually done using beetroot. Okay, and the first thing we need to know is that beetroot are the colour they are because they contain a pigment, like a dye, and that pigment is called betulin. Pigment called betulin. Okay, and that's what makes them the colour they are. And in this experiment, we're going to see the movement of this pigment through the membrane, or the lack of movement through the membrane, and that's what is the basis for this whole experiment. That's what makes the liquid go this reddy pinky colour. Okay. Um, the second thing is that it uses a piece of equipment called a colorimeter. I've got a picture of it here. So this is called a colorimeter. Okay, colorimeter. And what happens in the colorimeter is you put your little sample in here. So your sample is in one of these tubes. These tubes are called cuvettes. And you have to fill it up to the line. I don't know why they're all filled random amounts, but anyway, you fill that up to the line and you drop it into there. And then what happens is the machine shines light through your sample. Okay, so it shines the light through it. There's two settings. You can either have it on absorbance, which measures how much of the light is absorbed, or you can have it set on transmission, which detects how much light is transmitted straight through it, how much light passes through your sample. Okay, so that's what a colorimeter is, and you can absorb the amount of light absorbed by your liquid, i.e., how much doesn't come out the other side, or how much does come out the other side, how much is transmitted. So, in this experiment, obviously, we have to identify our variables first of all. So, we've got independent, and we're going to change dependent and control. Now, independent is a bit of a pain for this one because there's two different versions. So, the two versions that you can do are temperature, so you can put each one at a different temperature, like shown in the picture here, or you can do it with different alcohol concentrations. And you need to know both. Okay, but you would only do one at a time, so it's a bit confusing writing them both there. But let's say our independent variable was temperature. All right, so we always just change one thing. Um, so the dependent variable is going to come from your colorimeter, and let's say that we're going to do absorption. It wouldn't matter which one you did. Let's say we're going to do absorption of light from the colorimeter. Read that off on the colorimeter. And the control variables, things that we're going to keep the same. We should use the species of beetroot. We should use the same part of the beetroot. We should use, if we're using temperature, we should use the same alcohol concentration. If we put it in alcohol, if we were doing alcohol concentration, I'm not going to write that because it's a bit confusing. But if we're changing the alcohol concentration, you should keep them at the same temperature. Okay, the most important one, which is really tricky to keep the same, we'll talk about as we go through, the amount of time they're left. So, how much time do you give it for the betulin to leak out of the cells? That's really tricky. Okay, so let's go through the method. How do you do this experiment? So, I'm going to type this. So, the first thing you're going to need to do Number one, you're going to need to actually make your cylinders of beetroot. Now, it's really important that they're the same size. All right, so you cut your equal size pieces of beetroot. It's important they're the same size so they've got the same surface area. Okay, and the way we cut the cylinders is we use something called a cork borer. Cylinders of beetroot using a cork borer. And make sure they're all the same size. So you might do, for example, 20 millimeters long. So they all need to be cut with the same cork borer, and they all need to be measured to be 20 millimeters long so that they've got the same surface area. If we go back to here, actually, really important one we should have put on here is we should have put another control was the surface area of the beetroot. So each one needs to be the same size so it's got the same surface area. So we cut out our little cylinders of beetroot first of all. What do we do next? Now, you need to just rinse them under water first of all, because we're going to be measuring how much betylene comes out of the cells. And when you cut them, you're going to damage some of the cells and some of the beetroot juice, some of the dye, the pigment, the betylene is going to come out. There's going to be some betylene on the surface of your cylinder. So you need to wash that off so that you're only measuring the betylene that's getting released from the cells during the experiment. So you rinse off any betylene from the surface of the cylinders. Okay, so we're making sure our results are valid. So we're making sure that we're only measuring what we want to measure, we're only measuring the pigment that comes out during our experiment. The next one is that we need to put some water into our test tubes for them to sit in. So we're going to try to always use quantities. So we could say measure 5 mil, centimetre cubed is better, but I don't have a cube button on here. So 5 mil of water into 5 test tubes. And then each of those test tubes needs to be at a different temperature. So on here, look, we had the picture of tubes of water with the cylinder of beetroot in, each one being put at a different temperature. Okay, so measure out 5 mil of water into 5 test tubes, and then put those into water baths at different temperatures. So if we're doing the temperature one, obviously this is when it's your independent variable, try to always use 5 and try to always be specific, like actually name the numbers. So if you were doing concentrations, 5 concentrations, name them 20, 40, 60, 80, 100%, whatever. If it's temperatures, name the temperatures. Okay, so we could have 0, 20, 40, 60, 80, 60. Sorry, I can't make that very small. So be specific. 5, 5 values for your independent and specify them. Use numbers. It gets you more marks. Now they need to be left for the same amount of time. And this is where it's really, really tricky because it's hard to put your samples in the water at exactly the same time, but we're going to try our best. So add the beetroot cylinders. We should leave for, let's say, it doesn't matter, but use numbers where you can, 20 minutes to get to the right temperature. Because obviously straight away when you put it in, it's not at the right temperature. So then I'm going to add the beetroot cylinders to the water. I'm going to try and add the beetroot cylinders to the 5 tubes at the same time, if we can. Okay, so beetroot cylinders to the test tubes at the same time. Now you need to leave them for a certain amount of time to allow the betylene to come out of the cells. About 15 minutes is a good amount of time. So we can say leave in the water for 15 minutes to allow the betalain to come out of the cells. And what you'll find is that if I go back and look at this, some temperatures, hardly any pigment will come out of the cells and the liquid will still look clear. But then other ones, loads of pigment will come out through those membranes and the liquid will start to look really pink. All right, so leaving the water for 15 minutes to allow the betalain to come out of the cells, or not, depending on what temperature it is. It's really important now that we take them all out at the same time. What you don't want to do is take one out, measure it in the colorimeter, then take the next one out, measure it in the colorimeter, because then they haven't been in the same amount of time, and then your results are not valid. Results are only valid if you're only changing one variable, and if you leave them for different amounts of time, then you're also changing the variable of time, and so your results won't be valid. So we want to take them all out at the same time, all right? That's really, really important, all right? And we want to transfer the liquid to cuvettes, to these little square plastic pots that go in the colorimeter. Okay, so we want to take when I said take them out, all out, I just mean the beetroot. Take all the beetroot cylinders out, all of the beetroot cylinders out at the same time, and transfer the liquid to the cuvettes. Okay, so you take the beetroot out of all 5 and then put the liquid from all 5 into the cuvettes, ready to measure. Okay, then you use your colorimeter to measure. We said we were going to do absorbance, didn't we? The absorbance of each one, and then you plot the graph. So you put it in the colorimeter, you get your readings, and you draw your graph, and your graph is going to look like this. Okay, so at some temperatures you've got a really low absorbance, at other temperatures you've got a higher absorbance, and it should draw a curve like this. Obviously, you can also do different alcohol concentrations. So the difference there would be that you'd have, rather than 5 different temperatures, you'd put them in 5 different concentrations of alcohol, and you'd get a similar result. Okay, so let's have a look at what our results are actually showing us, what's actually happening. So, first of all, the higher the absorbance, it means that there's less light passing through the solution. So it means that there's more pigment in there. Okay, so you've got to be able to actually interpret what those results are telling you. So obviously the correlation on the graph overall, so we could say overall, increasing the temperature increases the absorbance, which means less light is passing through. And the reason why less light is passing through is because more pigment has gone through the membrane. So it's more pigment that's come out of the cells, the membrane is more permeable. Okay, less light is passing through, which is because there is more pigment in the solution, because more betalain has come out of the cells, which indicates that the membrane is more permeable. Yeah, so let me just go through that again. So as the temperature is increasing, the permeability of the membrane is increasing and it's letting the pigment out. So if you imagine what's happening is, if on a microscopic scale inside that cylinder of beetroot, we've got cells. Those cells are filled with betalain pigment, and the higher the temperature, the more permeable the membrane is, and so more pigment is coming out. And so we're ending up with way more pigment in the solution. Yeah, well, first of all in the tissue fluid, and then if you imagine that's in the cylinder, isn't it, of beetroot? And then obviously once it's in the tissue fluid, it can just come out of the cylinder and into the liquid. So what you had is a test tube with a cylinder of beetroot. Then within the cylinder of beetroot, you had cells, and the pigment has come out of the cells and out of the whole cylinder. And this liquid in here is turned red, hasn't it? Reddy pink colour. Okay, so the higher the temperature, the more permeable, increased permeability of the membrane. So more pigment came out, increased pigment, more betalain came out of those cells. Okay, and that's why this liquid around it got a darker colour. And that's why when you put that in the colorimeter, less light could pass through it because it was like dark red, dark pink. Okay, so overall, increasing the temperature. Oops. I think I could just rub that out. Let's just rub out the bit that's gone on the right. So overall, increasing the temperature increases the absorbance. So always reference, first of all, make sure that you reference the words or the axes. Yeah, so I've referenced the temperature and the absorbance. Increasing the temperature increases the absorbance. And then show that you know what it means. So that absorbance, if it's increasing the absorbance, more light's being absorbed, which means less light's passing through. Why is that happening? Sorry, it's a typing mistake there. Why is that happening? Because let's click. Because there's more pigment in the solution, more pigment has come out of the cells because more betalain has come out of the cells, which indicates that the membrane is more permeable. Okay, so that's what our graph is showing us that's actually happening. Now, if you're describing this graph here, this isn't a linear line. So you can always say during a description that it's non-linear, okay? It's not linear. So if you get a straight line graph ever, you always need to say that it's linear. If you get a shape that's not straight, you need to say that it's not straight. You need to say that it's non-linear. You could say this is an S-shaped curve. Can you see the letter S there? Like a letter S, kind of. So it's a non-linear graph, which is worth a mention. If you're asked to describe the graph, remember to reference numbers, so it increases from this number up to this number, and remember to do a percentage change, okay? So describe answers are always using those things. Now, if you're asked to say why it happened, if you're asked to explain, then we need to use the science. So everything we've talked about so far is really describing, and now let's look at why it happens. What's the science? What's the step-by-step reasons of why our graph is that shape? Okay, so first of all, what happens is as you increase the temperature above 0 degrees, what happens is the phospholipids get more energy. All molecules get more energy when you heat them up, don't they? So the phospholipids get, so we should start off, sorry, say as you increase the temperature, as you increase the phospholipids more energy. Okay, so what that means, specifically kinetic energy, more kinetic energy. What that means is that they move around more, and they're not as packed as they're not packed as tightly together. Okay, so they move around more. They are not so as tightly packed together. Okay, and that means that the membrane is more permeable. All right, it's easier for the betylene to get out of the cells. Okay, now if we think about the detail of the structure of a plant cell, we've got our cell wall around the outside, we've got our cell membrane, we've got our nucleus. We've also got this massive structure here called the vacuole. Now, the vacuole is actually where the betylene is. Okay, so it's not only the cell membrane that must have become more permeable, more leaky, it's also this vacuole membrane as well, which is called the tonoplast. Okay, so the vacuole membrane must also have become more leaky because that red pigment, that red betylene, has been able to diffuse out of the plant cells. Okay, and the hotter it got, the more permeable this membrane on the vacuoles become, the more permeable this cell membrane has become. Yeah, and so the betylene has been able to leak out. More permeable, so more betylene diffuses out of cells. And therefore the solution becomes a darker red, and that's why you got the higher absorption reading. Now, it's not a linear line, is it? It starts to plateau off. If we look at our graph here, it does increase as they get more kinetic energy and the membrane becomes more permeable, but it does start to plateau at the top. So if you were to keep going, it wouldn't keep up going up forever, because it can't really go up past 100 anyway. Okay, just there. Another thing to bear in mind is that the kind of steep increase might be not just because it's becoming more permeable, which might be the kind of first increase, but then actually the membrane might start to break down. Okay, so after probably about 45 degrees, probably about here, the membrane actually starts to break down and even more pigment will get out. Okay, so at first it's just that there's more gaps between the phospholipids, and then after about 45 degrees, above about 45 degrees, the bilayer starts to break down and it becomes even more permeable. The water inside the cell at that temperature as well, the water molecules get so much kinetic energy, the water would expand and put more pressure on the membrane. Proteins would start to denature and deform, like the channel proteins and carrier proteins, things like that, and they can't control what enters or leaves the cell, which in turn makes it more permeable. So you get this fast increase. Okay, so above about 45 degrees C, membrane starts to break down. We could include things like proteins denature, so channels or carrier proteins can't control what passes through. Water in the cell expands, damaging the membrane. Okay, and it becomes more leaky or permeable. All right, it's obviously then going to plateau at about 100% absorbance. can't go above 100, can it? Okay. Now, remember that this whole experiment can also be done with alcohol. So you need to know the answer for the alcohol version as well as the temperature version. Actually, before we go on to alcohol, I just want to talk about this graph here. You see the temperature graph goes down to 0. Sometimes they give you a graph that goes lower than 0. Let me show you what that would be like. So if you did it down to like -10, 0, 10, 20, etc., 80, whatever, the graph would actually go like this. So below 0 is also permeable. So above 0, we've got this increasing permeability that we've talked about. Actually, below 0, we've got increased permeability as well. Okay, below 0 degrees. And that often gets missed, but does occasionally come up. So below 0 degrees, the phospholipids have hardly any kinetic energy, obviously, because it's very, very cold. So below 0, very little kinetic energy. And therefore, sorry, I'm just, computer's running out of battery, just plugging it in at the same time. There we go. So below 0, we've got very little kinetic energy. So the phospholipids are not moving around much at all. They're very closely packed together. So lipids are still very close. So therefore, they can be very close together. You'd think that it wouldn't be permeable because the phospholipids can lie very, very close together. But the problem is that the channel and the carrier proteins allow movement through, increasing the permeability of the membrane. And worse still, what happens is that ice crystals form from the water and they pierce the membrane, making it really, really leaky. So ice crystals form and puncture the membrane. Okay, so it can become very leaky down at the low temperatures. Right, now let's go on to the temperature example. So, I'm sorry, the alcohol example. So if you do exactly the same experiment, but use alcohol, you get a graph like this. It get again this sort of S-shaped curve like this. Okay, so on this axis this time we've got alcohol concentration, and on this side we've got the membrane permeability. Okay. So what's happening here is as you increase the alcohol concentration, the membrane permeability goes up again. And the reason for this time you need to just learn a tiny little bit about this, is the lipids in the cell membrane get dissolved in the alcohol. Okay, as the alcohol dissolves the lipids in the cell membrane. All right. So then the last section is the evaluation, which is always very similar. It's worth us just going through the details that are specific to this practical. So first one, reliability. So remember, reliability begins with the letter R, repeat is with the letter R. So after your method, ideally we should always put repeat 3 times. Okay, you should discount any anomalies, don't include them in your average. Okay, so if you get two that are similar, one that's different, don't use the one that's different. And ideally, repeat it 4 times. So you've then got 3 good ones to take a mean of. Okay, then calculate the mean. Makes it much more reliable. Second one, reproducibility. And this is about comparing your results to another experiment that proves the same thing. So another experiment probably done by someone else, somewhere else on a different day with a different method. So another experiment giving the same results backs up your results. It means that your results are reproducible. Your conclusion is reproducible. You can reproduce it in another way. If you get the same results doing the same thing again, that's just more reliable. Okay, reproducible is a different experiment showing the same result. Next one's validity. I've tried to talk about this one a little bit as we go through. This is results are valid if you only change one variable. So things that make your results invalid is things like if you weren't able to get all the beetroot out at the right time. If you left some of them in for longer than other ones, then you weren't only changing one variable, were you? You also changed the length of time they were in for. And so therefore you were changing more than one variable. Okay, so validity is you only change one variable, and this means that you are answering the original question. Otherwise, you're testing something completely different. The next one is precision. So if your results are precise, it means when you repeat, you get similar results. Okay, if each time you repeat you're getting different results, then they're not precise. You should always be getting similar results. They're precise results. So accuracy is close to the true value. Means the real correct answer. So if it was done by someone who's really experienced in this kind of thing, who had the most amazing technique, the most amazing equipment, what is the exact answer that they got? Did you get close to that? Okay, so it's just about doing your experiment really carefully, doing your experiment really well, measuring things exactly, leaving them for exactly the right amount of time, them having exactly the right same surface area. Another thing we haven't talked about is when you take them out of the liquid. Oh no, sorry, that was a different experiment. Yep. So accurate, close to the true value, getting really good results. Next one is uncertainty. So whatever you're measuring with, it's half the smallest value. So here you're measuring with the colorimeter. Okay, so this can measure to the 1%. So half the smallest measurement is a percent, so that'll be half percentage. Okay, that's your uncertainty of your equipment. And the very last one is percentage error. So you can work out percentage error of any result, any single result, by doing the uncertainty divided by your reading. Okay, certainty divided by your reading. So, for example, if you got a reading of 80%, yeah, then your uncertainty is 0.5 divided by, yeah, 0.5 for this. We'll change that to 0.5 instead of half clearer. So 0.5 divided by 80, which is 0.8, is the 80% is 0.8 as a decimal. Okay. And the way to reduce your percentage error is by measuring always in larger amounts. Right, that brings us to the end of this one. And then we'll go on to the next point. Well done.